Hello and welcome to another lecture for Bio 111. Today's topic is sex linkage and recombination. Like always, let's take a look at how we ended the last lecture. So last time we talked about the fact that some but not all traits are dominant or recessive. And we talked about dominant recessive traits that are caused by alleles that are carrying one of two traits. There could also be multiple alleles for one gene. Two doesn't have to be the limit. Segregation of alleles happens because of independent assortment of the chromosomes into gametes during meiosis. So that means that during meiosis, the chromosomes are moving to opposite poles. And if you have one allele on one chromosome moving to one pole and the other allele on the other pole, then that is how they are assorted, how they're moved, how they are put into different gametes. I showed and discussed a Punnett square, and I showed how you can use it to draw out the possibilities of allele distributions and how it allows the visual calculation of traits in the offspring generation. So you can actually count out the distribution of the genotypes or the phenotypes if you write that down as well. We talked a little bit about mathematical ways of doing that. There were the rules of addition and multiplication. We talked about incomplete dominance as an alternative to the dominant recessive traits. I showed you how to do a test cross, at least in theory, which uses the phenotypic ratio of the offspring of a cross between an unknown genotype, so that's what you are testing, and a homozygous recessive tester, so that's what you're testing with, to determine the genotype of the unknown. So you can try those out in multiple ways, write them down in Punnett squares, in simplified Punnett squares, and then determine what must be the correct genotype of the parent. And then we looked at human inheritance and how that is used to study pedigree analysis, or how pedigree analysis is used to study human inheritance. All right, so today's lecture is about sex linkage. And so some traits are referred to as being sex-linked. And I just want to take a little detour here at the beginning of the lecture, just to sort of clarify a few terms. So let's first define what a geneticist usually means when they use the term sex. So biological sex refers to gamete size, really. So you might think, it refers to male or female, and in a way it does, but really the fundamental differentiation between sexes in organisms, not just humans, not just animals, not just plants, but any kind of organism, is really defined by whether or not the gametes that are produced by the one or the other sex is large or small. And so you have in many lower organisms, such as algae, you have a state that's called, or a phenomenon that's called isogamy. Iso means identical or the same, and gamy means use of gametes. So isogamy means having the different gametes, but of the same size. Many algae have iso and isogamous inheritance. And then there's anisogamy, and that means that there is a small and a large gamete. So in many cases, that is then referred to as the male or the female, because the male has the small gamete and the female has the large gamete, the egg. And so in order not to conflate what sociologists might think about when they talk about femaleness or maleness, you could also, in biological terms, just think about this as the sperm producer, that is usually referred to as the male, or the egg producer, that is usually referred to as the female. And then in biology, there's all kinds of diversity. There's also hermaphrodites, which produce both. So you could have egg producer, sperm producer, or sperm and egg producer. So if this is the definition of biological sex, then there's the other term that's often conflated with it, and that is the term gender. So gender refers to how someone knows themselves to be. It's an identity. It's not the same as the biological sex. And so we just want to be sure that we keep those two terms apart when we talk about genetics or we talk about biological systems, as opposed to how humans are or feel or interact with each other. And then another term I want to point out here is the term of intersex. What is intersex? Intersex is so intersex refers to people who are born with any of a range of sex characteristics that may not fit the notion of a binary male or female body. So in other words, anything that's in between. And there are multiple ways how that can occur. So in many cases, we just think of binary ways. We just talk about males or females. But we want to be aware of the fact that there's also intersex, where humans or animals can be born on a continuum with a variety of sex characteristics. All right, so with that in mind, let's talk about sex chromosomal inheritance in humans. So most humans have an X or a Y or two X chromosomes. And what's typical is that females have two X chromosomes and males have an X and a Y chromosome. The X chromosome is much larger than the Y chromosome. And if you think back to meiosis, the way that they get distributed to different cells, to different gametes during gamete production, just like the other chromosomes, the body chromosomes or the autosomes, they align with each other. They align with each other during metaphase, in the metaphase plate. Well, if two chromosomes need to align, they have to be homologous, they have to be very, very similar. And that's what homologous chromosomes are, right? They are the same chromosome, the same gene arrangement on the chromosome, same way, same number of genes, but there might be slightly different versions. So those are the different alleles. Now, how does that work with an X and a Y chromosome that are so different from each other? Well, the reason why they can align with each other is because their tips actually do have regions of homology. So you see these red tips here, they can align with each other during meiosis, and they can make sure that the X and the Y then segregate into separate gametes. All right, so when we think about assortment of sex chromosomes into gametes, what we normally have is a 50-50 ratio of male to females. And if you look at this from a chromosomal perspective, then here is why that is that way. So in meiosis, a female has two X's and will produce in her eggs one X chromosome or another X chromosome, while meiosis in a male will result into a 1-to-1 ratio of producing a gamete with an X chromosome or a Y chromosome. So if the Y chromosome from the male and the X chromosome from the female fuse, then you have an XY offspring, and so that offspring will be male, and you will have two of those possibilities in this Punnett square. Or the other 50% chance is that you have an X from the male and the one X from the female, or the other X from the female. And of course, that is also, in both cases, then female. So if we look at chromosomes and sex chromosomes specifically, and the traits that are inherited that lie on sex chromosomes, then there are some special ways to think about inheritance of traits that are located on sex chromosomes. And the reason for that is the fact that they are not that the chromosomes are not completely homologous. So let me show you an experiment that was done in the 1920s Thomas Hunt Morgan, was working on flies, on fruit flies. And he was crossing flies with different mutations to each other. And one experiment that has sort of become famous and that is described here in your book is the one where he crossed a red-eyed female with a white-eyed male. So you can see that the females are a little bit bigger in these flies than the males. You can see if you look very carefully, you see the red eyes here and you see the white eyes here in the male. So when those were crossed together, you would see in the next generation red-eyed females and red-eyed males. All of the progeny had red eyes. And then in the next generation, white eyes reappeared, but only in the males. And that is curious, right? Why would it not also reappear in the females? All of the females had red eyes, and among the males there was a 1-to-1 or 50-50 ratio. So this conundrum is what Morgan studied, and what eventually led him to the insight that this had to be a trait that was that resided on the X chromosome. And let me show you how you can sort of deduce that from looking at sex chromosome segregation. So if you have an affected male, so this is an affected male, and that male has an X chromosome and there's a mutation on that X chromosome, then that mutation will be passed down in his sperm carrying the X chromosomes, but not in his sperm carrying the Y chromosomes. So if a male passes down this trait, then all of his offspring that inherit his X chromosome, that means all the females, will inherit an X chromosome with this trait and an X chromosome from the mother with, in this example here, with the wild type trait, so not with the mutation. And if it's a recessive dominant correlation, so recessive dominant trait, then the dominance of the X chromosome from the female will take precedence, and so therefore none of these females will show the trait. But in males, there is no second option, right? The second sex chromosome doesn't carry the information that is carried on the X chromosome. So the Y chromosome simply doesn't code for this. It does not have that gene. There is no eye color gene on the fruit fly Y chromosome. And so that means that the males, all of the males, will have to inherit the Y chromosome. Otherwise there wouldn't be male. And so they will inherit the typical X chromosome from the female that has the eye color, of the female. So a male with an X-linked recessive trait will have heterozygous carrier daughters and normal sons. These are the sons, and these are the carrier daughters. So a carrier is an organism that carries a mutation but doesn't show the trait. So it's masked by the other chromosome. So I'm just writing this down here for you to notice. Carriers are defined as a heterozygote female not showing the trait because of the second X chromosome. Okay, so now let's take a look at the same cross, except that we're now going to look at the genotypes as well. So remember how in the example of the peas, with Mendel, we had the large letters and the small letters, the capital letters and the small letters. By convention, geneticists have come up with their own nomenclature depending on the organism they work with. And so it was customary back in the days, and still is, for flies to use a different nomenclature. So you don't have big W and a little w for the white mutation. You have a W plus and a W minus. And so here the convention is that the plus is the unmutated typical trait, and the mutant trait is the minus trait. Sometimes that's not so clear to tell which one is mutant and which is normal, right? So it's just two different traits. So if we look at this cross and we look at the genotypes, we see that the red-eyed female has the W plus. So two chromosomes, both of them carrying the dominant trait, and the male has the W minus, but only on one chromosome. And then since he doesn't have a second X chromosome for which we could denote whether it's W plus or minus, because there just isn't another version, we just write down here that the male has the Y chromosome. So the Y chromosome is neither W plus nor W minus. So if we then get the offspring from these, so W plus and W minus, that would be this possibility, or W plus and the Y chromosome, right, that would be the other possibility, then we will get in both cases red-eyed organisms, female flies and red-eyed male flies. And the reason, of course, is that this is dominant, so the W plus will take precedence over the W minus, and the W plus in the male will take precedence over nothing on the Y chromosome. And then if we cross these with each other, so basically sibling cross these, then the offspring will be W plus and W plus is one possibility, or this W minus and that W plus. So that will be this combination. In both cases, it will be a female, and in both cases, it will have red eyes. For the males, the possibility is the W plus and the Y, W plus and the Y, and so that will be a red-eyed male plus the Y chromosome, or the W minus, which comes from the mother, and the Y chromosome, which comes from the father, of course. So the W minus and the Y will then create the same genotype as the grandfather, which was wide-eyed, and so this male grandson will also be wide-eyed. So I'm writing here as the things to remember from this cross, that the male has only one X chromosome and will show the recessive trait, that the male can only pass on the one X chromosome that he has, and so all daughters will receive the X chromosome that he has, regardless of whether it's the recessive or the dominant trait, and that carrier females in the F1 will pass on recessive X chromosomes to 50% of their sons, because they have two that they can pass down to their offspring. So if the carrier mates with a recessive X male, then both male and female offspring will segregate 50-50 or 1-to-1 for the recessive X chromosome. So let me show you how that works. So this cross is a little bit different. So we start with the same red-eyed female with two dominant alleles, and we start with the same male, has the Y chromosome and has the recessive W minus allele. And we will get the same as on the previous slide, carrier female that has the W plus, W minus. But now in this one, we're not doing a sibling cross. We're crossing this with a wide-eyed male. So basically the same genotype as the grandfather. And so what you will then get is the combination of a W plus, W minus here, W minus here, and then the W plus Y chromosome or the W minus Y chromosome. And of course, the two with the Y chromosomes are males, and you can see that they will inherit either the plus or the minus, 50-50 chance, so they will be white-eyed or red-eyed. But in this case, the females are also also have the possibility of showing the mutation. And that is because that chromosome, the X chromosome with the mutation that comes from the father, together with the chromosome from the mother, from the carrier mother, can also produce the homozygous recessive phenotype. And so therefore, in this case, white eyes appear in both sexes at a ratio of 1:1, or 50-50, 50-50 between these and 50-50 between those. Okay, so that is sort of the gist of what X-linked inheritance is about, or sex chromosomal inheritance is about. There's also the possibility of inheritance that is linked to the Y chromosome. We'll come back to that a little bit later. There's a lot fewer genes on the Y chromosome. That's why there are a lot fewer traits that are inherited via the Y chromosome. But we'll come back to that in a little bit towards the end of the lecture. So what Morgan's team also was able to show with these experiments in flies was that genes sit on chromosomes. So your book tells you a little bit about how that is really not something we are too surprised about these days. But back then, they didn't know that. They didn't even know what DNA was. They didn't know what the nature of DNA was. But what they did know is what chromosomes were. They were able to see chromosomes in the microscope. So you squash a cell, you see that there's stuff inside, and it looks like discrete little bodies. That's why they're called chromosomes, some for body, and chromo for color. And they're called color bodies because you could add little chemical droplets of chemical solutions to them, and they would stain. They would take on the color of this chemical solution. And even if you washed it off, they would take on that stain. That's why they are called chromosomes, color bodies, because after rinsing them, they would keep the color. So how did they find out about this? So they did experiments, and how good geneticists are, they don't assume that just because they find weird results that it's necessarily their fault. To be frank, when I do an experiment and things don't work and I don't understand them, I most likely would throw it away and do another experiment. And maybe these folks did that as well. They did another experiment, but they wrote down the numbers. They did it a third time. They found it again. And at that point, they start scratching their head and they say, maybe it's not me. Maybe it's something for real that I see here, and that I can explain if I just come up with the right hypothesis. And so this is one of those examples. So let me show you what they did and what you would normally assume, and then what they didn't think and how they interpreted it. So normally, chromosomes separate during meiosis, right? So we know that in cell division, these two chromosomes will be moved to the poles. And if these two chromosomes are X chromosomes, then this will result in segregation such that this gamete will produce an X gamete, and this will produce the other X gamete. So if this fuses with another X, it will be a female. If this fuses with another Y, then it will be a male. So normal chromosome separation results in each egg containing a single X chromosome. And then in a cross with white-eyed females, so this is a white-eyed female, normal chromosome separation results in female progeny with red eyes and male progenies with white eyes. So you have the red-eyed male here. Here's the W minus, W plus. That would be a red-eyed female because she doesn't have a Y chromosome, and because the dominant W plus allele is the one that codes for red. And then the males would be, they would have the Y chromosome, and they would always get one of those two alleles, which are the same alleles in this case when you use a white-eyed female. And so he would always get the W minus and the Y chromosome. So you would assume that all really. XY males receive their X chromosome from their mother. Now, these folks in Morgan's lab didn't always observe that. And so when they found that the ratios didn't come out the way that they thought from what I showed you on the previous slide, they came up with this hypothesis, and then they tested the hypothesis, and in the end showed that yes, indeed, this is a way to explain the outcome of the results that we very infrequently but sometimes see. So what is it that they saw? They saw that when they made the cross, they got infrequent individuals among their normal individuals that they saw, where they saw the red-eyed females and the white-eyed males. They found some white-eyed females and some red-eyed males. And so one way how they decided this could be explained was if the X chromosome failed to disjunct normally, in other words, go and separate so that one chromosome would be on one and the other chromosome would be in the other pole, and then lead to eggs that were X in either one of them, but not XX in one egg cell and 0, no sex chromosomes in the other. But if that happens, if you have non-disjunction, then you would end up with 2 chromosomes on one side and no chromosome of that kind on the other side. And that would then lead to white-eyed females that have 2 W minuses here on this side, right? So they get both of their X chromosomes plus a Y chromosome. And on the other side, they would have the red-eyed males that have the red chromosome from their father. So that suggested to them that on the chromosomes, if chromosomes physically not disjunct during meiosis, and that affects the traits that they see, that something on the chromosome, physically on the chromosome, had to be responsible for producing these traits. And of course, today we know that those are the genes, and that those genes are located on the chromosomes. But that was new to Hunt Morgan's team in about 100 years ago. All right, so let's take a look at some pedigrees that we can analyze, that we can look at, human pedigrees, with respect to some of these X-linked mutations that we just discussed. So here's an example of an X-linked recessive mutation, the example of color blindness. So people who cannot see the green dots in this picture here, if you are not colorblind, then you will see a number 5 here where I'm pointing with the mouse. But if you are, then you just see sort of a grayish, reddish, orangish type of mix. And this trait, relatively frequent trait in human populations, is passed down on the X chromosome. And so if you have a pedigree, the way that you would analyze it to determine whether or not this is a dominant or recessive, whether it's an autosomal or a sex chromosomally related trait or inherited trait, is as follows. So you could look and you could see that, so we have a trait where for the X-linked recessive trait, most affected individuals are male. So if we look just overall, we see that the ones that are affected seem to be, in this case, all male. And then we look for telltale signs where heterozygous daughters of affected males, so this is a male and she is heterozygous potentially here because she doesn't show the trait where she's unaffected, but she's the offspring of this particular male that was affected. If she has affected sons, like here, then she must be a carrier. And then the offspring of an unaffected male is usually not affected. So we don't have any offspring here, but we see that the sisters of an affected male can also have affected sons. So here are affected sons, and she's a sister of an affected male, right? So here are the affected males. She's an unaffected sister, and she can have affected sons. Now, this is the only case in this family where we see that, because this is not a daughter of this grandfather. She marries into this family, right, and and produces these daughters. Okay, so these would be indications for a pedigree that shows an X-linked recessive mutation. Here's another example of an X-linked recessive mutation that is frequently used as an example to explain or to show X-linked recessiveness. It's sort of an interesting example, and you might want to spend some time just looking at it. If you're at all interested in royalty, European royalty, you'll see that these are essentially here's Queen Elizabeth II and her husband Philip, and their offspring, Charles and his brothers and sisters, but Charles is the only one who's named here. He and Diana have kids and so forth. And you don't see any affected males here, but this is an example of hemophilia. This is a disease where a genetically inherited disease passed down on the X chromosome, where you cut yourself and you're bleeding. There isn't a certain gene that is required for making the platelets stick to each other so that they can seal the wound. So this trait is very frequent in the royal family, and you see that there's been a lot of inbreeding in the royal families of Europe, because royalty, you don't find royalty very frequently, and so people were married to cousins and related family members, because that was one way how to have influence. If you marry, the king's daughter to somebody else over in a different country, then there's influence that you get that way. And so you can see how these genes with that kind of inheritance spread throughout the entire larger family of the kings and queens of Germany and the UK, Russia, and Spain. And in this case, you see the affected males. These are all affected males. And then that some must be proven carrier females. So for example, in this case, she must have been a carrier, even though we may not have genotyped her, because Alfonso the 13th was not affected, yet his sons, two of his sons, had the trait. And so that's a way how you can analyze this. Okay, so let's move on to the next topic. So the next topic is about gene linkage. There's a bit of a problem with the nomenclature. So, gene linkage comes right in this discussion here in your book after sex linkage, and both of them are called linkage. Yet what is meant is something quite different with respect to linkage. So I'm going to try to keep those things separate for you. Sex linkage, meaning that there's a gene that is linked because it physically resides on a sex chromosome, as opposed to gene linkage, where you have two genes that are linked to each other and they do not necessarily have to be on the sex chromosome. And that's what this next part here is about. So this is interesting, because when you know that genes are linked to each other physically because they're next to each other, then you can use that information. You can use some information. I'll show you some experiments in a second that you can do, and you can test how closely these genes are linked to each other, how closely they must be on the chromosome to each other. Let me show you that right now. It's probably a little bit nebulous the way I explain it. So we're thinking about how closely genes are physically located on the chromosome. So if we do this experiment, and your book chooses to use an experiment where the genes that are physically linked to each other happen to be on the X chromosome. Now that doesn't have to be; they can be anywhere. But this one happens to be also on the X chromosome. So you also have to account for whether it's the Y or the X chromosome that's inherited, even though that doesn't really matter so much for the discussion of gene linkage. All right, let's walk through this. So we have the parental generation, then we have the F1 generation, the F2 generation. We have a trait, we have two traits. One is the eye color, the W plus W minus, so that's the white and the red. You see them here, red and white. And the second trait, so just another gene, a completely different gene that we're looking at, is whether or not the wing has a cross vein. You see these little lines here, these veins that go to the wings. If you don't have this particular gene called CV, cross vein, then you don't have these cross veins. So the wing is probably a little bit less stable. Maybe the flying properties of this fly are affected. So we have a cross. Female is homozygous for both non-mutant alleles, for the red eyes and the normal wings. So that's the non-mutant, the red eyes, and the normal wings. And the male carries both mutant alleles for the white eyes and the cross-veinless wings. So this is the white and the cross-veinless wings. Now, the way that fly geneticists write this is they do this on the top of the line and the bottom of the line thing. And that is one chromosome versus the other chromosome. So remember that we're now looking at two genes on the same chromosome. So the way that this is drawn is that on the top of the line, we have the one chromosome, one of the homologous chromosomes, and on the bottom of the line, we have the other homologous chromosome. So these two are together on one chromosome, these two are together on one chromosome. And in this case, since this is on the X chromosome, happens to be in the X chromosome, then we also, for males, must have a Y chromosome there. So in the first generation, the first offspring generation, the progeny females are heterozygous for both mutant alleles. So let's look at that. We have the red-eyed, cross-veined females, and you see that they're heterozygous because you have the plus on the top and the minus on the bottom. So that's one of the chromosomes, and that is the other chromosome, right? So that makes sense. If you get this chromosome from this mother and this chromosome from the father, then in the next generation you will have this maybe on the top and this on the bottom of the way that it's written here. And for the males, it must be the other way around, right? So they get the Y chromosome is on the bottom, and then either one of these two chromosomes is the one that's shown here on the top. So in the next generation, they decided to just show the males. And in fact, there's a typo here. You see that it says only males shown here, but in fact, they also show female. They meant this to be down here. And so in this case, we're only showing males. That's not important for the discussion of gene linkage, because this just happens to be on the X chromosome. It makes it a little easier because we don't also have to account for what's happening on the second chromosome. Maybe that's why they chose this example. So let's take a look here. These are all males. They all have a Y chromosome on the bottom. They then turned out to be this way. This is what they looked like. Plus. So this is red-eyed and cross-veined males. So the two dominant traits, or the two recessive traits, wide-eyed, cross-veinless. So that is the recessive trait here, right? Wide-eyed and cross-veinless, while this is red-eyed and having the cross-veins. But then you see something that is surprising. You see that there is a red-eyed individual with cross-veinless. So they have a plus and a minus on the same chromosome, and they have a minus and a plus in this particular wide-eyed cross-veined male. So what does that mean? Well, that must mean that if these two genes were physically located on the same chromosome in the grandparents, but now they are no longer physically located on the same chromosome, they must have been broken and rearranged. They must have been recombined. The two chromosomes must have broken and fused and recombined. And when does that happen? Well, of course, during meiosis, when we have crossing over. And that is exactly what can happen. And when I told you about what Mendel did in Mendel's experiments, I said at the beginning, sort of on the side, Mendel was kind of lucky in that he chose the right traits and the right experiments. Well, what I meant with that is that he never got this kind of slightly messy experiment because he happened to choose traits that were on separate chromosomes. He didn't have this problem where chromosomes were on the same, traits were on the same chromosome. So let's look at the chromosomal level and see exactly what I mean with that. So if you have, just like in the fly example on the previous slide, if you have genes that are more tightly linked to each other, then they will be less likely to recombine or to split apart. So there are two ways how these two genes, A and B, and they could be the red wide eye, so let's say red here and white eye here, and the cross veinless here and the cross veined here with a little b. So this is how they could be recombined during meiosis. So we have here the two homologs, and they align during metaphase plate. So crossing over in prophase 1, that's when they start to align, that's when they start to when they start to exchange, and then after metaphase, of course, in anaphase is when they separate. So in meiosis, we have the physical exchange of the chromosome parts, right? So in other words, crossing over happens during prophase 1. And the way that it's drawn here is that the crossing over happens in between, physically in between gene A and gene B, right? So if that happens, then this chromosome now has a recombined, it's really a recombined chromosome. It has a capital A here and a small b here, when it started out having the two dominant traits next to each other. Now it has a dominant and the recessive trait next to each other, and it swapped that with this other chromosome. And then, of course, when it goes to the next phase where the sister chromatids separate from each other, then we have our four gametes. They move into the four gametes, where we end up with four different types. We end up with this type, so the big A, big B. Now remember that these letters, when I say big A, big B, these are two different genes, right? So they can be together, or the little a, little b can be together, or the big A and the little b, or the little a and the big B. These are the recombined ones. See, these were the parent ones, so the blue or the red. And these are now the recombined ones. But we also have non-recombinant ones. So we have the ones that did not recombine. So this is just like the parental one. So one of the two chromatids becomes a chromosome during anaphase 2. And so therefore it is a non-recombinant chromosome that can be inherited, while there's also recombinant chromosomes that can be inherited. And that's this one or that. So your book says here, the overall frequency of recombinant chromosomes is a measure of the genetic distance between two genes. And I want to come back to that in a second. Why do you think that is a measure of the distance between two different genes? The overall frequency. So think about that for a little while. We come back to that in a second. I want to show you this other example here on the bottom, where crossing over also only takes place once on the chromosome, but it just happens to take place in a different location of the chromosome. So in this case, where the recombination occurs below the two genes that we are interested in, the recombination doesn't change anything with respect to the combination of the two genes that we're following. These are still recombinant chromosomes, but they're not recombinant with respect to A and B, with respect to genes A and B. You see that here we have blue A and red B. Here we have, even in the recombinant chromosome, red A and red B, or blue A and blue B, right? So these are non-recombinant for the particular genes. Okay, so let's come back to the question of why genes that are more closely physically located to each other on the same chromosome might recombine more frequently. Well, if you think about this, if A were to sit further here and B were to sit further over here, then there's a bigger stretch in between where you have the chance, it's random chance, right? Where this happens is completely random, relatively speaking, completely random. And so it's more likely that there will be a recombination event, as it's called, a crossing over event between A and B if they are further apart from each other. If they're really, really close to each other, if they're right next to each other, it's very unlikely that the crossing over will happen just right in between those two, right? If they're further apart, then there's just more room in between where the crossing over could happen. And so, therefore, the overall frequency of recombinant chromosomes, in other words, the likelihood that recombination occurs just between the two genes that we're interested in, is a measure of genetic distance between the two genes. And that is what we're going to use now to do some calculations with respect to where two genes might sit. So let me just write this down here and say that if A and B are far apart from each other, it is more likely that crossing over between them occurs. Right? The further they are apart from each other, the more likely that this event happens to happen in between them. And when that occurs, when the recombination occurs in between them, like it does over here, then you are unlinking the two genes from each other and you're producing recombinant alleles. Those are then just called recombinants. And that is the process of unlinking two otherwise linked genes. And so that's what I said earlier. We have these two different ways of discussing or using the term linkage. Here we're talking about linkage of having two genes being physically so close to each other that they are almost linked to each other, as opposed to linked to the sex chromosome, which is sex chromosomal linkage. Different term different way of using the term linkage. Okay, so in this case, we now wanted to do some experiments. Let's do some experiments, and of course these were done for us. Experimentally obtained numbers of offspring of these flies, where these crosses were done and where the offspring were counted, the F2 offspring were counted with respect to their traits. And so they found that out of these total of 795 flies that were analyzed, they found that 357 had this combination, 52 had a recombinant combination with a big blue A and a little red B, 45 had a recombinant combination with little a, big B, and then there were 341 that had the parental one, the non-recombinant little a, little b. So the majority of events showed that the chromosome was passed down to the next generation intact with respect to A and B. So the recombination, the crossing over, did not happen between A and B in the majority of cases. But there were some, and not that few, that recombined right in between those two. So we said earlier, the further these guys are apart from each other, A and B, the more likely it is that recombination will occur between them. So I say here, it appears to be more likely that genes A and B are inherited together than would be expected if they were independently assorted. So what does that mean? That means that if they were independently assorted, and remember that Mendel coined this term of independent assortment. Well, completely independent assortment would mean that they are on different chromosomes, that they are always, there's no linkage, that they are independent with respect to each other, whether they go into one gamete or the other gamete. But that's not the case here, right? If you sit on the same chromosome next to each other and your chromosome is sorted in one direction, if it's not broken and recombined, there is no independent assortment. But when they are on different chromosomes, there is independent assortment. Now, under what conditions could A and B be independently assorted? Well, of course, under the condition that there's recombination occurring in between those two genes. And so when that happens, you get this outcome. This is the outcome that you get for the recombinant flies, where that occurred in between A and B, and this is where it did not occur in between A and B. Now, the further these two genes are apart from each other, the more likely this should happen. And so you can use those numbers by first adding them all up and then adding only the recombinants and dividing them by the overall number to determine what the frequency is of recombination. And so if you add these together, this is 97 divided by 795, you get, comes out to be about 0.12, about 12%. 12%, 92 of 795. And so that number is the number that gets put on something that's called a genetic map. Let me show you this genetic map. So if you have a gene, here's the W gene, and here's the cross-variant gene, and the recombination frequency, so the frequency with which they are separated from each other on the chromosome because of crossing over, is a certain percentage that we just calculated. Then that is the recombination frequency, and that is the distance, just by definition, with which they are put on a what's called a genetic map. So here's a genetic map. Here's the distance between the W gene and the CV gene. We calculated out to be about 12.2%. And so you see that there's a difference here between W and CW of a total of 12.2%. And now if you do this with other combinations, if you look at the phenotypes of the gene that's called yellow, or the gene that's called ruby for ruby eye color, and you look at the combination frequency between those two genes, so that means you set up another entirely different experiment. You have different flies with different mutations. You make the crosses. You look at how frequently they become recombinant versus parental in their phenotype. And you do the same kind of analysis, and you say, oh, the difference between those must be 7.5. That's the recombination frequency, the percent of the recombination frequency that we calculated here, right? That is the value, the 12.2%. Well, if we did this experiment again and we got numbers here for two different genes, then we could do the same information for yellow and EC, or W and RB, and EC and CV, or any of these combinations. And they should always give us a relatively similar result, because if we do this just often enough, hundreds of times really, then the frequency with which crossing over should occur between these different genes should always be the same. And so then you can put the genes with that number, and that number is sometimes referred to as centimorgans. Morgan, just the name of the guy in whose lab all of this work was done. So this is just to honor him, that the distance on a genetic map is referred to as centimorgans. So this would be 12.2 centimorgans, or 12.2%. And the book says here that across sufficiently short distances, so when we're just looking at relatively small distances on a chromosome, the map units are additive. So we can add these up. Thus, the expected frequency of recombination between W and CV is this plus this plus this, if we didn't have information from an experiment that just uses the CV and the W, which we did though. So with that, you can then also verify that. So this is a genetic map. And these kinds of experiments were done, dozens and dozens of these experiments were done so that different types of traits could be mapped onto the gene, so that researchers would know whether they are really close together, whether they're on the very top of the chromosome, whether the very bottom of the chromosome, whether they were on the same chromosome or on different chromosomes. Okay, so let's, we probably have another 15 minutes left or so, so maybe take a quick break, come back, and then we'll talk a bit about some more modern approaches to using gene markers and making maps to determine certain traits in humans or other organisms. Okay, so these genetic maps can be done with what's called markers. I find the term marker difficult. I've always had a hard time understanding what a marker is. So let me try to put that in terms that might help. So a marker could be a gene, right? A marker could be a gene, but it doesn't have to be a gene. A marker is simply any region on the chromosome that is specific, where you have specific information. And the way that you can have specific information is when you have different alleles. So you can say, I mark the chromosome at a certain allele, and that could be a gene, for example, for white or red eyes. And then that gene, the red and white gene, becomes your marker. And since you know that these alleles are versions of the same gene, and that the versions arise because of mutations, and the mutations are in fact just simply nucleotide changes, the mutation essentially is the marker, right? So essentially the mutation provides the reason for why this is a visible marker. Now, we talked about genomes a little bit, and chromosomes, and how many genes are on an entire chromosome, and we said that chromosomes are full of genes, but they are full of a lot more DNA that are non-genic. So there's non-genic or intergenic DNA, and that intergenic DNA, that can also be used as markers if there's a difference between two different chromosomes that you want to distinguish. So we talked about it in the context of VNTRs and SNPs. And SNPs are the single nucleotide polymorphisms. And so SNPs happen all over the genome. They're everywhere. There are thousands and thousands of SNPs that, for example, the human genome has. If you're looking at a particular trait, let's say a disease trait, and you would like to know where this trait is located on the chromosome, you can't really do the types of experiments that Morgan did. But what you can do is you can do pedigree analysis. Now, pedigree analysis requires quite a bit of information from quite a few people. One thing that you can do these days, and we'll get a little bit more into that later on in the class as well, is by doing what's called an association study. So you associate a phenotypic change with a marker that you may know, that you may have some information about. So let's say you have information of markers across the entire genome of a human. And let's say you have 1,000 markers. You know the physical location. Now, why would you know the physical location of these thousands of markers? Well, now that the genome is fully sequenced, you basically run the entire chromosome set, the entire genomic, all of the chromosomes, through a sequence analysis, and you write down all the letters. Now you have the entire genome. Now you can compare entire genomes of humans, of individuals, with each other. And you can say, oh, I see very clearly there that in individual A, or 1, I have this particular sequence, a G and a C on the other chromosome. And in very few individuals, I see that there's an A and a T. Just happens to be so. And then you go another 100 nucleotides further on, or 1,000 nucleotides further on, and there will be another SNP. And as I said earlier, and what I want to emphasize again, these SNPs, for the most part, are outside of genes and do nothing, right? They do no harm. They are not associated directly with producing a phenotype. But they are there, and you can use them, and they just happen to be numerous and different between different people. And so if such a SNP is located close by, relatively close by, a disease gene, it doesn't influence the disease gene. It has nothing to do with the fact that this gene carries a disease. But if you know the identity of this particular SNP, whether it's a GC or an AT, and you know whether the person has the disease, which is relatively easy to know, right? If it's a physical phenotype that you see, then you can take the genomic information and you can say that, well, interesting, in almost all cases, I had a person with this disease also carry this particular SNP. That means there must be some sort of association, there must be some sort of linkage between the SNP that I know and the gene for which I only know that it confers some sort of a phenotype. I don't know what the gene is, I don't know exactly where the gene is, but I do know where this SNP is, and I do know that whenever I see that SNP, or almost always when I see that SNP, that person also has the disease gene. And when I see this SNP, I do not see the gene. So in this case, AT is associated with a non-mutant phenotype, GC is associated with a mutant phenotype. Well, then, if that is the case, then that means these two traits, let's call them, are linked, right? This is not a gene, so I can't call this gene linkage. But I can say this SNP is linked to this disease gene. Well, then, if we just said AT is not linked, is linked to the non-mutant version, then how is this explained? Well, that's explained because every now and then there will be crossing over, right? There will be crossing over. And in this case, there was crossing over right between those two. And that does not happen very frequently if the location of the gene or SNP we're interested in is relatively close to the disease gene. So let's see what your book says. It says, in an analysis of 100 chromosomes, most show an association between a mutant allele of a gene and a GC SNP, suggesting that the mutant allele is located near the SNP. Now, if there's no association, if you take a SNP and there's no association, then this should be completely random, right? You should see on average the same combination. So in this case, there's no association between the disease gene and the SNP. So the location of the disease gene in relation to a specific SNP, is a random specific SNP that you chose and you thought might be related to this gene, cannot be determined. So that means this SNP doesn't tell us anything about whether or not a person might have this disease gene. But if I know the identity of the SNP in person X, then I can say with a certain percentage of certainty that more or less likely that person also carries this disease gene. And so that's how markers, which you can easily nowadays with whole genome sequencing determine for every person, can tell you something about traits even if you don't know the genetic identity of this particular trait. So markers are any true allelic locations on a chromosome. They can be a gene, they can be SNP, they can be a VNTR. Markers can but don't have to be the mutation causing the reason for causing this mutation, the trait of interest to be useful for mapping the trait within the vicinity of the marker. So here we see that they can be useful for mapping the gene to the vicinity of the marker because they are linked. Now, we do this with thousands of markers, and if we're lucky, we find one that is linked, but that's all we need, right? That's what we would like to do. And so you do a lot of work, but in the end, maybe you find one marker of the 1,000 that you have, and then you can use that marker in the future to determine whether a person, even if you don't know the identity of the gene, you can't look for that itself, whether that person is more or less likely to have that particular disease gene or that particular allele. So that's what mapping looks like today. And then, previously I showed you what mapping looked like in Morgan's time. Okay, let's try to wrap this up here quickly. I said that there are Y-link traits and that they are passed from father to son, and I wanted to show you an example where you see one of these pedigrees. So in a pedigree where the Y-linked traits are passed on, you see that only males exhibit this trait. So this is a gene that presumably is on the Y chromosome, and so therefore no female will ever have it. Females do not inherit, nor do they transmit the gene. And all sons of males with a trait also show the trait. All sons of males with this trait, here's the male with the trait, all sons will have it, because that father has only the Y chromosome to pass down, and so all of his sons will have to have that Y chromosome. Otherwise, they're not the typical XY male. So using that information, people use something that's called a haplotype analysis. So haplotype just means haploid genotype. So haploid means just half, right? If you are diploid, you're looking at both of the homologous chromosomes. If you're looking at only one of them, you're looking at the haploid genotype. And so the haplotype for the Y chromosome is easy because there is only one Y chromosome, right? There's never the other one. So your book explains a little bit how the Y chromosome can be used over time to determine heritage. So when I say determine heritage, what that means is where in the world, where on Earth, this person's ancestor came from. So back in the days when the world was populated, people were first evolving, humans were first evolving in Africa, as we now know quite well. And you will learn more about evolution of humans if you take Bio 112. So they were located in Africa, and then they started moving out, and some of them moved out to Europe, and some of them moved out to Asia, and then from Asia, some of them moved down to Australia, and some others moved over the Bering Strait that was frozen at the time to Alaska, and then from Alaska down into the Americas. So how do we know that? Well, we know that from archaeological findings and digs and so forth. But we can also tell that from looking at the relation of our genome, and the Y chromosome is relatively easy to understand, even for somebody who's just starting to understand genetics, and why you can use that. So let's take a look at the Y chromosome and how mutations over time can create different haplotypes, and how you can then say, well, if I have that haplotype, I must be more closely related to an ancestor that also had that haplotype. So here's our most ancestral haplotype of the Y chromosome. And then at some point, there was a first occurrence of a mutation. So there's a red letter is now a mutation. You see how that A changed to a G. Let's say this human here was in Africa, where humans first evolved, and the mutation occurred still in Africa. And so these were people that lived in Africa, and there's another mutation here. These are still people that lived in Africa. And each unique combination of nucleotides represents a different Y chromosome haplotype. So this is a different haplotype from that. It could be that this arose from that if this male had a mutation that caused the change from the T to the A. But this error should mean potentially thousands of generations in between. So now let's assume that the person with this haplotype, with this Y chromosome genotype, had offspring. Let's say this person moved out of Africa and had offspring, and some of his offspring went in one direction, let's say into Europe, and the other went into Asia. And so now they're starting to still accumulate mutations in their DNA that are, irrelevant to function. It's just intergenic DNA that accumulates these mutations. So not a problem, but you see how now they are separate. These two lineages are separate, and you can follow their separate lineages because all of this male's offspring now have the same haplotype, the GA and the A here, GA and the A here, GA and the A at the end, and so forth. While the offspring of this person, of this male, continues to have this haplotype, right? This mutation doesn't go away. It's always GA at the beginning. Actually, that's the same here. but this person doesn't have the A at the end, while this person does. So now you can start to distinguish between people, males in this case, for the Y chromosome, carry this particular haplotype versus males that carry this particular haplotype. And then if you find that maybe these are the ones that split into Southern Europe, and these are the ones that went into Eurasia and Northern Europe, and these are the ones that stayed in Southeast Asia, and these are the ones that went over the Bering Strait into the Americas. Then you can, if you have current DNA, you can say, okay, I have something that looks like this haplotype. So my ancestors must be most closely related to the person that was in this location of the globe. So that is how haplotypes work with respect to ancestry analysis. So mutations in the Y chromosome in red accumulate over time. Separate allele combinations can be used to distinguish individuals or populations. So you can distinguish somebody who comes from a population that has this haplotype versus one that has this haplotype. And oftentimes when you read about this, even in the popular literature, these haplotypes of Y chromosomes are then used to show the geographical origin, and they're shown by the percentage of people with a certain type of haplotype, with a certain kind of haplotype. So these pie charts will say, let's find one that's, maybe this one is 40%, 40%, and 20% of people in Scandinavia have either the yellow haplotype, the green haplotype, or the purple haplotype. And this yellow haplotype, people with that particular genetic sequence on their Y chromosome are much more frequently found in Northern Europe or Northern Asia, and much less frequently found in the Americas. So from this kind of analysis, you could then say, if I know about this, and I know where the haplotypes originally were from, and now I pick a person's genome, and I sent my genome to heritage. I'm not sure what it's called. There's one that's called 23andMe, and there's a number of them these days. Anyway, so I send it there, and they take my money and they do my sequence analysis, and then they tell me, well, your analysis, your haplotype looks most likely most similar to the red haplotype, and there's hardly anything else. Then you can say, looking at the map, or they will give you the answer, of course, they will say that means that your ancestors with this particular haplotype who lived in this particular region of the world, that your ancestry is in this particular area of the world, as opposed to maybe one that has a very different haplotype that is hardly ever found in other places. Now, of course, these days, in the age of jetting around the globe and mixing a lot more than people used to, just maybe as late as 150 years ago or so, hardly any mixing was going on, or very little comparatively was going on. You can use that information to determine where ancestors originally lived. Okay, so one last thing, and that is that organelles are also, they also have DNA, and the reason why they have DNA, chloroplasts and mitochondria, is because they originally were microbes that were engulfed by other microbes. We heard about that earlier in the class. So chloroplasts have DNA and mitochondria have DNA, and they don't do the regular kind of meiosis and mitosis that our nuclear DNA does. But they have to be passed on from cell to cell as well. And so they get passed on by cell division. And so whatever cytoplasm happens to be in the cell on the left carries the mitochondria that happen to be in that cytoplasm at that time, and the ones that were on the right go into the other cell. Turns out that mitochondria in humans are passed on from the mother to their offspring, never from their father. And the reason is because the cytoplasm for the sperm, there's hardly any of it there. And so there aren't any mitochondria in the cytoplasm of the sperm cells, but the egg cells are large, and so therefore they carry the mitochondria, and they can be passed down to from mother to son or daughter. Doesn't matter, right? So you can see here a pedigree for a particular trait that is inherited on a mitochondrial DNA, and you can see that it comes from mother to all of her offspring, mother to all of her offspring. mother to none of her offspring because she doesn't have that trait, because it's the male that has the trait, right? So he inherited it from his mother, but he cannot give it to his daughters or sons because the inheritance goes through the mother. All right, enough for today. Let's summarize this quickly. I started out by saying that biological sex is determined by gamete size. We talked about the way that we can refer to sperm producers and egg producers, and that how that is usually referred to as male and female. I discussed briefly how gender, the term gender, refers to a self-defined identity and has nothing to do with biological sex. So we need to keep those two things apart when we talk about sex or gender or certain traits of humans, because there is a continuum and there's a lot of diversity and variety. So then we talked, with that caveat, about sex chromosomes carrying genes. We called them sex-linked genes. We said that in animals with an XX-XY sex determination, that recessive genes on the X chromosomes show up in XY individuals. And I said that genes can be linked to each other on any chromosome, and that linkage is not the same as sex linkage. And then we talked about how linkage can be mapped, how it can be used, how analyses for segregation patterns can be done by doing experiments where flies are crossed with each other or any other organism, and that from the number of recombinants versus non-recombinants, you can calculate the relative distance of genes and put them on a genetic map. And then in the end, we use that a little bit to see how we can use this type of marker analysis to look for disease genes, and how we can use marker analysis to look for traits, or maybe just molecular traits that we don't see physically, such as the haplotype of the Y chromosome, and how that can then be used for determining ancestry. And then in the end, I told you a little bit about mitochondria and how they are maternally inherited in humans. Chloroplasts are also inherited monoparentally, not biparentally in plants. But the species differ. Some species have male parental, others have female parental inheritance. The point that you need to remember about this is that mitochondria and chloroplasts do have a genome. It does have to be inherited, and that mitochondria in humans are maternally inherited. All right, and with that, I let you go, and I'll see you in class.